This is a study that we're very passionate about. Both myself and my colleague Dr. Leikau have had a personal interest in cancer, and as neuroscientists we came to this question from a different angle. And so we started this project from a very simplistic idea, was that different variable natural history, how much of that related to an individual's interaction with their environment, and how much specifically did the brain have, what sort of input did the brain have on the outcome of that cancer. And so what we did is really looked at a very simple paradigm which we and others have used to look at the brain's plasticity, or the brain's interaction responsiveness to the environment. And that is really taking animals out of their controlled housing into what we call an enriched environment. An enriched environment essentially is typically a larger space, a space in which there's larger social groups, and there's also activity, places, toys, running wheels, various places for the animals to hide and run and be more physically active. So it's a combination of both physical and social stimulation.
Yeah, we're very interested in how the environment influences on the brain, but we're also very interested in how the environment have influence on the cancer growth. So firstly, we use highly aggressive melanoma cells to test this idea. So we house the animals in the enriched or control housing for 6 weeks, followed by implantation of melanoma cells. And we found a very remarkable 80% decrease of melanoma mass in the enriched animals. We then generalized this paradigm to another well-defined marine cancer model, MC38 colon cancer. And in this experiment, we tried to investigate whether the enrichment worked even after the cancer have already been established. The growth rate of this cancer has been significantly decreased by enrichment, even this intervention has been delayed. So third model we used is an APC mean mice, which is a spontaneous model. Very similar to the implantation models, 6 weeks of enrichment was highly effective in reducing both the total number and the size of the intestine polyps. In order to tease out the mechanism of this anti-cancer phenotype, we targeted hypothalamus. The brain area is critical in the regulation of metabolism and neuroendocrine and immune function. We found a significant specific upregulation of BDNF in hypothalamus, which was not seen in exercise-only animals. In order to prove whether BDNF is the mediator linking environment to cancer growth, we used the AAV vector to deliver BDNF gene to the hypothalamus. The overexpression of BDNF in hypothalamus mimics the enrichment effect, including a sharp drop of leptin level, enhanced immune function, and a 75% decrease in the melanoma mass. Then we move on to investigate the potential peripheral effectors in this regulatory network. So the most consistent and profound change is a sharp drop of leptin level. Leptin is a fat hormone which is predominantly secreted by the white adipose tissue, and its production is suppressed by the sympathetic nervous system through beta-adrenergic receptors expressed on the fat cells. We found an upregulation of the 3 beta-adrenergic receptors expression in fat cells, as well as increase in norepinephrine levels in the fat. When this sympathetic drive was blocked by beta blocker, enrichment was no longer able to induce a sharp drop of leptin or inhibit tumor growth. We implanted a leptin-releasing liposome to the animals and housed mice in the enrichment. The exogenous leptin offset the enrichment-induced leptin drop, and the enrichment could not decrease tumor growth in this setting. So taking all these things together, we propose a mechanism which is the environment switch on BDNF expression in the hypothalamus. BDNF activates hypothalamic sympathineuron adipocyte axis, HSA, and HSA shuts down fat hormone leptin expression, then inhibit tumor growth.
What we've shown here is that an animal's interaction with its environment, putting animals into more complex environments, a more challenging environment both socially and physically, has a marked impact on the growth of cancer. The hypothalamic sympathoneural adipocyte axis, and that axis is activated by this complex environment. And in activating that pathway and inhibiting the production, the release of leptin, you have a dramatic effect on cancer. We already knew that obesity is a risk factor for cancer, and the state of your fat mass actually has an impact on prognosis. But what's most important about this study, and I think what's provocative here, is that these environments in which animals are more stimulated, they're certainly not free of stress. In fact, when we measure the stress hormones in these animals, the stress levels are a little bit elevated. So this complex environment in which they have more challenges both socially and physically drives this pathway, this axis in the body that has a direct input on cancer growth. Thank you.